Hi everybody, and we are going to continue on talking about the membrane today, and we are going to focus on membrane transport, or how things get in and out of a cell. And so that is our big question for today. How is it that materials can get across a cell's membrane? So there's a couple problems that we have to keep in mind when we look at this. The first is that phospholipid bilayer that we talked about yesterday is hydrophobic, and so hydrophilic materials, or anything that mixes really well with water, is polar. Those materials don't get across a cell easily. And we also have a problem with large materials, because if they have to get through that phospholipid area, some molecules are going to be too big to actually easily fit through the membrane. So two mechanisms we're going to talk about are passive transport and active transport. Passive transport is something that requires no cellular energy for the movement to occur. And so there are three types that we're going to look at. We've got diffusion, osmosis, and facilitated diffusion. Now, diffusion is when you have a net movement of atoms or ions or molecules that move down a concentration gradient. So they're going to always move from high concentration to low concentration. And this is what we have in our example right here. So we have molecules of dye on the left side. Then we've got this membrane, and this is all happening in solution. And so what's going to happen is those molecules are going to start to diffuse or move through the membrane until they're at equilibrium. And so here you can see that it's about equal concentration on both sides of this membrane. Now what's interesting is once they're here, they're not going to stay in these exact locations. For every molecule that you have while at equilibrium that would move from the left to the right, you will also have one that would move back from the right to the left. And so that's when we say that it's at equilibrium, there's no net movement. So for every one that moves, you'll have another one move the opposite direction. Here we have diffusion of two solutes, so again it's the same thing. The purple ones are going to move or diffuse from the left over to the right, and again the yellow are going to also diffuse, and they're going to go from this right side over to the left until they both are at equilibrium. And so equilibrium, concentration is equal on both sides, and again, no net movement of materials, but you will have a move in both directions equally. Factors that could affect diffusion: concentration of the solute, temperature, pressure, particle size, mixing. Those are all going to affect how things diffuse in solution. Next we have osmosis, and this is the diffusion of water. And so here we have water moving from an area of its high concentration to an area of its low concentration, and this is still passive transport, so we have no energy being used. When we talk about osmosis, we also talk about tonicity, and so this is going to be the concentration of water relative to a cell. And so we've got isotonic, meaning the cell and the solution are at the same strength, hypotonic, meaning below, and hypertonic, meaning above. And so when you look at a hypotonic solution, here we have a cell in a hypotonic solution, and so we know that there's essentially a solution on the inside of a cell as well, because it contains proteins and all kinds of molecules and the organelles and lots of stuff on the inside of the cell. And so when that cell is in a hypotonic solution, or a solution that has fewer solutes than the inside of the cell, water is going to want to rush in. Now with an animal cell, if water rushes in, the cell will start to swell, and it could actually lyse or burst. Here we have the same thing happening with a plant cell, so we've got water rushing in. However, because the cell wall is surrounding a plant cell, it's not going to burst the same way an animal cell would. Here we have an isotonic solution, and so essentially this cell is at equilibrium. Iso meaning the same, so the cell is in a solution of the same strength, and so you've got water moving in, but you have the same amount of water moving out. So this is a normal animal cell. And then here we have a hypertonic solution, and so when you put an animal cell in a hypertonic solution, that means the solution strength outside the cell is higher, there's more solutes, and so that water is going to want to leave the cell in order to dilute the surrounding area right here. So the water is going to leave, the cell will then shrivel up. Now again, in a plant cell, it's going to look a little bit different, because that cell membrane is going to be attached to the cell wall, and so it's going to look like it's kind of stretching in, and so we say it's plasmolized at this point. Alright, so just to review, isotonic, it's when the cell and the water are equal in solute concentration, so equilibrium, no net movement of water, but some water will still move in and some water will still move out, and so we have no change in cell size. With a hypotonic solution or a hypoosmotic solution, in this case the cell has more solutes than outside, and so the cell's water then is lower than the outside, and so what's going to happen then is water is from the outside is going to rush into the cell, and that's why cells could burst. And then with a hypertonic solution or hypoosmotic solution, the cell has less solutes than outside, so the cell's water is higher. And so water is then going to move out, and that's what causes the cell to shrink. All right, next we've got facilitated diffusion. And so this is when you have a transport protein that's going to help materials move through the cell membrane. Now, even though a protein is being used, this does not require ATP because you still have materials moving on their own from an area of high concentration to an area of low concentration. The protein is just there to make it a little bit easier for the molecules to move through the cells. So here's an example. You've got high concentration on the outside of the cell, and so these molecules want to move into the inside of the cell, but they could be too big or they could be polar, and so they can't actually get through the actual phospholipid bilayer, and so the protein just kind of gives them a nice little, it's almost like a little sidewalk for these molecules to move through easily, because they would get trapped here. They can't get into through that phospholipid bilayer. Aquaporins are an example of this, and these are channels for osmosis. So these are channels that help water move in and out of a cell because, again, water is not going to like that hydrophobic region of the phospholipid bilayer. Next, we have active transport, and so this is going to be movement that does require cell energy. And so we've got three different examples. We have carrier-mediated transport, endocytosis, and exocytosis. So carrier-mediated transport is just a general term for the active transport of materials against a concentration gradient. And so this is when you see materials move from an area of low concentration to an area of high concentration, which you obviously wouldn't expect because we just spent all this time talking about diffusion. But we do have a few examples, and so we've got the sodium-potassium pump, we have hydrogen ion pumps, and then we have co-transport. So this is the example of the sodium-potassium pumps. And so you can see here that you've got sodium on the inside of the cell, and it's binding to this protein, and you can see that ATP is being used here. It's giving some energy. The phosphorylation then is going to cause this protein pump to change its shape, and it's going to release the sodium to the outside of the cell, and this is where sodium is actually higher. So we would expect the sodium to want to stay in the cell because it's got a lower concentration here. But with this being active transport, we're going to pump it to where it's already much higher. Once this happens, now we've got this protein that's open to the outside of the cell, and so we have potassium that's going to bind, and then it is going to be released inside the cell where the potassium is already higher. And so this is just a very common example, and again, this is the sodium-potassium pump. And so again, moving sodium ions out of cells while moving the potassium ions in the cells. Next, we have the electrogenic pumps, and these are also called proton pumps, and they're going to create voltages across membranes for other cell processes. And so here we can see we've got hydrogen in a low concentration inside the cell, but a high concentration outside the cell. So you're going to have energy from ATP, and these hydrogen ions are going to get pumped through this protein into the outside of the cell where that concentration is going to continue to build up. Another example is co-transport, and so this is the movement of hydrogen ions that's going to allow other materials to be transported into the cell as hydrogen diffuses back across a membrane. And so an example here is sucrose transport. So we have this same proton pump, so the hydrogen ions are going to get pumped to the outside of the cell. Once they're here, they're going to actually diffuse back in through another protein, and as they diffuse back in, they will essentially take this sucrose with them. So both the hydrogen ion and the sucrose will diffuse back into the cell then through this co-transporter. All right, next we have exocytosis, and so when you look at this word exo, sort of think exoskeleton, the outside, cyto, cell, so exocytosis is a way to move materials to the outside of the cell. And with exocytosis, you're usually going to be moving bulk materials outside of the cell. An example here would be when a cell secretes enzymes. With endocytosis, endo meaning in, and cyto meaning cell, so this is when you move bulk materials into cells. And so we're going to look at a couple examples of endocytosis. Pinocytosis, which is when you're moving liquids, phagocytosis when you're moving solids, and then receptor mediated uses receptors to catch just very specific kinds of molecules. So really quick right here, we are looking at our endoplasmic reticulum, and the endoplasmic reticulum, then the Golgi apparatus, and so you've got these Golgi vesicles with materials that are ready to be secreted out of the cells. So the vesicle is going to carry these materials to the cells. You can kind of see here that the cell is butting up, or I'm sorry, the vesicle is butting up against that cell membrane. And it fuses with it, and then it's going to release all of these materials to the outside of the cell. And so this is an example of exocytosis. Here you can see vesicles that are being formed. And so essentially what you guys need to know is membrane structure. You guys should be able to discuss the various methods that cells move materials into and out of their cells through membranes. And you're going to have to be able to solve some problems in osmosis. So we will spend some class time looking at isotonic, hypertonic, hypotonic solutions and how they affect cells. All right, and we're actually going to start with one quick problem in osmosis for your lecture question. So I want you guys to send me a little note and explain why a patient in a hospital would never be given an IV of pure water. All right, thanks guys.